Okay. Hope you all can see my screen.
Yes, Madhu, it's visible.
Okay, fantastic. Thanks a lot. Thanks a lot. Russ and everyone, thank you so much for participating in this webinar today. I really appreciate it. So in the next, as Simona said, in the next 45 minutes or so, I'll try to take you through the current topic, opportunities for AI in a personalized medicine. So during the presentation, I'm going to talk about a different topic. The first is to set the stage, specifically, talk about the drug discovery and development paradigm, and emphasis on the valley of a death, specifically looking into the phase 2 and 3 conundrum, and establish the justification and why do we need the patient stratification specifically at this juncture of a phase 2 and a 3, and also that if there is a huge failure in a phase 2, and what would be the current challenges, why there is a failure, and how the stratification is going to be beneficial if we apply to the very good design of the phase 2. So we'll talk a little bit more about it. While doing that, we can also talk more on what are the different ingredients in AI ML oriented algorithms are required, starting from the data, the analytics, and the model. And then post that, we'll switch the gears, specifically talk about the 2 different case studies, and both are in the clinical area. The first case study is we're trying to elaborate more on trial data benchmarking, specifically the phase 2 trial data, using that and the benchmark using that data and predict the certain algorithms, certain biomarkers to utilize in a phase 3 clinical trials. We will do that. And a second case study, we are going to use the phase 1 data to establish the classifiers or the markers, specifically whether given an individual is qualified for the inpatient or outpatient when we are subjecting them to the CAR-T treatment. So with these 2 case studies, I wanted to emphasize not just the utility of the here. In the first case study, I like to go more details about, the whole 9 yards of the different ingredients, what kind of things we should be doing with the data, what are the different approaches, the analytics we should look into it, and then what's the importance of the scientific justification of the outcome. So I'm going to take you through all of those different aspects of the overall journey of the data modeling. Okay. First and foremost, you all are familiar, right? The drug discovery and development paradigm, which starts specifically from the initial stage of the target identification, which comes under the basic research, to all the way to the pushing a compound into the shelf of the pharmacy. Right, in this entire journey, we all know that it's a very long process, and the time taking requires quite a bit of money. End of the day, the percentage success is the less than 1%. So between these 2 different major phases, I would say the preclinical and the clinical, there is a component called valley of death. The author, all the way you see at the bottom at the reference here, the Sehan. I think this gentleman really put the concept of the valley of death in his paper very well. I really intrigued and picked this up. So this valley of death is at in between the basic research and the clinical research, where this is where there is quite a bit of a failure is happening. So let's focus a little bit more on this. What is this valley of death, right? And why the individual clinical trials are getting failed here, specifically at the juncture of the phase 2. So what's happening at the phase 2, right? When we look into the phase 2, there is a number of papers are published where exactly the failure of the clinical trial is occurring. right? Each one that are on the left side, there are 7 or 8 different studies conducted, taking the clinical trials from in a different time range. And then it concluded that, what is the failure or a success rate? I would say in this case, the probability of a success, that's what is being represented here. So, and of course, each one has a little bit of a variation here and there. So that's quite normal when you're looking into the different time period, different methodology people have used. But the most interesting thing is it's a phase 2 to phase 3. The success rate of the phase 2 is relatively low compared to the success rate of the phase 1 or the success rate of the phase 3 to registration. So an average, it is ranging about a 30 to 35%. And similarly, on the right side, if you look at different therapeutic areas, more or less the phenomena is the same. And the success rate of the phase 2 is, again, it's ranged anywhere from 20% to the 40%. Of course, at the bottom, the psychiatry, for the right reasons, it's only 23.5%. Point, we should note that point. But all in all, the success rate is very low. The bottom line, the message we wanted to convey here is the success rate is very low. Now the question is, what are the different factors contributing to this low success rate in a phase 2, right? When we can initially conduct the phase 2, what happens is this is the first time an investigational drugs is administered in a patient, not the healthy individuals, in a patient. And the patients would have a different heterogeneities, their different variation, their body composition, the way the response is totally different. So leaving that aside for a moment, let's look into the other factors. What are the other things are contributing to this failure, right? So first and foremost is the pharmacology of the compound, right? There may be a low bioavailability of, there may be dose-related restrictions. There is efficacy may not be there. The safety issues might be there. So the pharmacology of the compound is very much important. And then the next thing is did we identify the right target or not, right? Many things works in animal experiments, but when it comes to the human, things may not work as it worked in animal experiment for a variety of reasons. Translatability is going to be the major challenge. Species to species difference would be there. So these first 2 factors, we can always, circle back. We can do a little bit of iterations, down the road, and can easy to address. But the most important aspect is when it comes to the third aspect about the study, selecting the study population, this is where the patient heterogeneity comes into the picture. And if you select not the right patients, you will not get the right results. Hence, the efficacy would have a huge impact on the efficacy, and obviously the phase 2 will fail. So it is the utmost important that we should have a very good selection criteria into the patient. But how do we select those? How do we establish the very good criteria? And how do we stratify the right patient to the right clinical? This is where the right biomarker is required to identify the right patient. So then moving into the next slide, let's learn a little bit more about the patient stratification, right? There's a subtle differences, right? I mean, extremely on the left side at a 10 o'clock position, right? This is a typical traditional medicine oriented clinical trial. This is where you give the one dose, one medicine for different heterogeneous population. In some people it works, some people it doesn't, some people you will have adverse events. So this is, we all know there is nothing new here. That's a traditional medicine. On the extreme right side at 2 o'clock position here, the patient stratification can be done based on a certain points, right? whether the disease subtypes, whether the clinical feature, whether the biomarker. In this case, what we are doing is we are targeting the certain patient population to get the proper response. So hence we are saying this is the patient stratification. At the bottom, precision medicine. In this case, we have to utilize the right parameters for the right patient. So this is a completely precision or a personalized medicine where we have to make sure that whether we use the biomarker, whether we use the companion diagnostics, etc., that make sure the drug should work for all different individuals with the different characteristics, with the different heterogeneity. So there is a slight variations within the patient stratification. Of course, we all know the advantage of the stratification. Specifically, we improve the efficacy and approval rate increases. The number of patients required to enroll into the clinical trials will be less. Reduce the cost. There are many advantages in terms of when we use the right patient stratification methods, when we classify the right patient for the right medicine, there is a plenty of opportunities. So now the patient stratification, we all know that if we just do a simple PubMed or a Google, many diseases, this is being currently, many therapeutic areas, this is currently being used. So this is not necessarily a novel phenomena, but I think implementation is going to be a challenge. Mostly the oncology is taking the prime therapeutic area that where people are using a lot, followed by the CNS and psychiatrics, etc. So with this, I just want to ask a quick, a question. Sumana is going to a flash about 3-4 options for you. The question I would like to ask you here is, what is the challenge in applying AI ML to the patient stratification? Is this going to be dealing with multiple data modalities, whether it's the insufficient data or interoperability in the biological context, or finding the right algorithms? So there could be multiple answers, but pick your best answer. So you can, it takes a couple of seconds, then we can move on to the next page. Okay. Okay, so let's move on to the next one. So these are in this slide, we are going to talk about the right ingredients for the patient stratification, right? So we start with the heterogeneous patient population within a disease. Then for these heterogeneous population, there is a number of different things that we measure, whether it is the immunological factors, the phenotypic related information, biochemical, hematological, all of this coming from the EHRs, the companion diagnostic, lab results, genomics, proteomics oriented results, many different things. So technically, we will be using all of these different factors we use and consider after identifying the right ones to stratify the patients who are responders and non-responders. So we will be playing with some of these ingredients in the subsequent case studies. Okay, so the next thing is just some fundamentals about it, at least in my personal opinion. What are the different things when we are applying the patient stratification oriented methodology? What do we do, right? So the first and foremost step is a data processing. The second step is feature selection and modeling. Third is the results and the interpretation. When we say the AI/ML model building, machine learning model building, we think that that is going to be the major one, but that's not. Actually, the model building is technically takes about 20% of the effort. But whereas the data processing is the major portion, that's about 40% of the effort is required for the data processing. If we do not have the right data, or model ready data, it's going to be the garbage in and garbage out. So hence, to have a right model, to have a right outcome, as a scientist, we should be spending as much time as possible to select the right data and also to process that particular data so it can be easily pushed into a particular model building exercise. Then once the model has spit out the outcome, right, the most important thing is interpretation, scientific justification. We just don't simply believe in saying that, okay, my model gave the ABCD results, I'm going to utilize or implement in the clinical trials. No, that's not going to work out. So we really need to make, in order to, when we do this scientist to scientist talk, we have to explain that, why this particular biomarker might be more relevant to stratifying the patient. So it's a 40-20-40, a principle one should be using when we are doing the patient stratification. And the right side pretty much elaborates a little bit more. This particular piece talks about the data processing, the labeling, the training data, learnings, etc. Where the middle is the algorithms and model building, the bottom is evaluation, analysis, and the interpretation. But if you just take the right side ones, there are different modeling, feature selection modeling approach. It could be a simple classification, it could be clustering, or it could be regression oriented models. It depends on whichever the one want to choose. I mean, everybody has a different variations, preferences, and also the data may also demand certain approaches one should be using it. So with this, let's spend some time on two different case studies. I'll take you through more details about what's happened in this case study, how did we use, what did we do, the entire the entire story we will be talking about through these case studies. Okay, the first case study, as I mentioned before, this is a trial, phase 2 trial data for benchmarking, establishing the right trial design for the phase 3. This is specifically for an interleukin antagonist. And so for all practical purpose and also to preserve the confidentiality of the customers, our partners, so we have masked several things, and also in some cases, certain type of a data might be representative. Please bear with us. When we see, we can ask the questions, but some we can reveal, some we may not be able to, but I'll try to give you a story without divulging too much here. Okay, so in this particular case study, the objective is we have taken the legacy trial data for an IL agonist in autoimmune indications. Then our goal is to identify the potential biomarker for the better response, and that we have used the different approaches, as I mentioned, the data formatting, the building the algorithms, and outcomes, etc. Then that certainly was useful in improving or optimizing the clinical trials. Then what are the challenges we are addressing while we are doing this, right? The data to start with is extremely heterogeneous, different types of the data. In one end, you may have a lab results, on the other end, you may have a SNPs in the expression information or the methylation. So it's different types of data sets. You may have a continuous variables. So it's totally, heterogeneous. So with that, so when we started this particular case study, we purposefully wanted to take this as a challenge. When I say challenge, we really wanted to see if given a problem, how different people or different teams will approach in a different fashion, right? So the problem is the same. The way we approach, way we implement might be different. And the outcome, we are hoping that it would be the same. If the outcome is more or less the same, at least through multiple teams, then that outcome is more meaningful, more real outcome. Specifically, when we are talking about this model things, right? Everybody wanted to do their own way. Hence, we purposefully subjected, given this particular challenge, to the 6 different teams internally. At the bottom, and we also recruited to this particular task about different capabilities, different individuals who has the different expertise and allocated to these 6 different tools. We created in a war room kind of a situation to address this in a very short amount of a time, more maybe a 4 to 6 weeks at the most. So it is not just a case study, it is more of a try and test how people will behave when they receive a particular data set. What are the different things one would do? What are the approaches they will implement? What kind of outcome they will get for the same problem? So that's the diversity we wanted to test in this particular case study. Okay, so the logistics are 6 different teams. They would be reviewing the progress on alternative days. In majority of the, many cases, when they are massaging the data, then formatting the data, everybody, discussed with each other collaboratively. They made the decision. Once the data massaging is done, at the model building stage, they 6 different teams become independent to each other, and they performed, they implemented on their own. And finally, all the 6 different teams gave their final recommendation about what are those markers, they got out of their own model. So that's the way we approach this problem. But end of the day, we took those 6 different results and kind of, put it together in Excel spreadsheet and see how much of overlap, who got what, those kind of analysis is done. So with that, let me take to the next slide. Here is a very quick the roadmap of how we have done. The data journey started with the data, and then the step 1 and 2. This for about at least 7 days. It is mostly to do with the data normalization, conversion of the data, addressing the missing values, doing some univariate analysis, normalization. So all of those things are done specifically in 1 and 2. And because since we are putting everything here, so at each step we presented what are the different things people have done. I'm just simplifying it. 1 and 2, the data formatting. 3, 4, 5, 6 is basically the model generation. 3, 4, 5 is the model generation. 6th is once they generate the model, at step 6, we have given an independent set. Okay, this is called 800 sample set. 800 samples to build the model. 160 data sample to test the model. So in the 3, 4, 5, whatever the initial data we have, they can use whatever they want and build, But once the building is done, once they establish their own way, I'll give you a little bit more details. You will understand when we are more on 3, 4, 5. But at a 6th, now everybody is back to getting the same data they're supposed to use. And then 7th and 8th, more of a coming up with the final features. Then 8 is more biological interpretation, rationale development, and the presentation, etc., etc. So this is the overall nutshell in terms of the roadmap we have created for this particular task. So the work phase is distributed into stage 3. 3 stages. Stage 1 is the data processing. Stage 2 is the data analysis. Stage 3 is the potential features and signatures, and finally the recommendation. Okay, I'll take you through the stage 1 very quickly. So here I'd like to spend a little time here, so to just give you the overall steady design. So this entire thing, the data what we have is the phase 2 clinical trial data. It's an anonymized patient level data This particular data came from two different studies, independent studies, which were done in a different times using the same drug with some variation in terms of how they approached, but the same drug with the same dose. And for this particular phase 2 clinical trials, there were about 5 different endpoints they have measured to understand the efficacy of those the drugs. Okay, and in these two studies, the treatments are, one is the interleukin antagonist, second is a TNF inhibitor, a third group is a placebo. Within the interleukin antagonist group, there were 2 different doses, 150 milligrams and 300 milligrams. So this is in a nutshell, here is the experimental design. On the right side, we measured from those individuals, there are about 2,000 plus subjects, 48 clinical parameters, 28 biochemical diagnostic parameters, 98 SNPs with the genotype information. So we are using these variables are the parameters to build our models, and our goal is to identify the biomarkers from these 170 plus parameters to identify the suitable one which can stratify the responders and non-responders. So that's the overall study design. With that, the first step is data processing, right? Now we have, when we look at these about 170 plus variables are there. There is variables are in a number of different ways, right? First and foremost thing is the data formatting. And this is the very crucial in my opinion. There is so much of a, sometimes it is a text, sometimes it is a numbers. So first and foremost, we converted that wherever the text is into the binary conversion to the yes or no, into the 1 to 0, etc. In the overall 172 parameters or variables, there are a few columns where there is some overlapping information is there. So we reduced, or we merged and reduced this. And duplication columns, the meaning is same, the information what we get out of that particular variable is same, we have to remove that. In certain columns or certain parameters, there is a missing values are there, we address the missing values, and we establish a certain criteria. If this missing value is less than, greater than so many number of patients, we may want it to remove, or sometimes we might have imputed. So multiple things we have done. In other cases, we looked into these columns and see how much of a variability is present. For example, I'm just picking an example from a thin air. If a particular column has all S's, a variable represents only S, and there is no, because it's completely, there is no variation. Hence, there is no point in having that particular column in our data model. Then there are few variables like ethnicity, race, country information were ignored. Then in some cases, certain labels, outcome labels, which are the endpoints, we have combined those endpoints and created an artificial endpoint. Specifically in this particular case, in this particular case study, we have created an artificial endpoint representing some kind of a cumulative scoring mechanism. So with all of these things, our first step of a data formatting, data processing, removing the noise, all of the things we have done, it's a very, very important step. And just for your information, this is the total distribution of the number of patients, right? There are total 2,078. I mentioned, right, we used the 2 different studies, the study 1 and the study 2. And here is the total. Within the study 1, you have about 2 different doses, 150 milligrams of this, 300 milligrams of a drug. In one particular study, we have a TNF inhibitor, another one we don't. Then the placebo. So with this, there are altogether 2078. And I believe there is a typo here, so it should be 221 rather than 2,221. Apologies for that. Okay. with that, now what we did is we started with, I mentioned, right? We started with the raw data, right? When we start with the 2078 samples, 170 features, 5 outcomes. For those 6 teams we have selected, we gave a free hand. We told these 6 different teams and saying that here is the data, guys, you take it. And you go ahead and do whichever the way you wanted to slice and dice the data. Right? They went ahead, a particular team might have used the entire thing, 2078 samples and all features. And they have used both drugs and all the doses. In some placebo, they have utilized in the model building. So in other teams, they created 3 different versions of the variations, the data. Version 1, the 2078, 162, the version 2, 333 samples, and so on and so forth. And how did they create? They had their own justification, why they took a small number of samples, why they took the 160, a small number of features, etc., etc. That way, another team is simply, they wanted to build the models separately for the different doses, right? So, with this, when we said that, you go ahead and whichever the model you wanted to use, whichever the AI/ML methods you wanted to use, so it's up to you. So once they build everything, then finally what we have done is this 800 model set is the testing one, right? So with this, the approach is independent approach. Once the process is established, they should be using this separate data to process through those models which they build, right? So that's the intention of this. Okay, so before we just jump into the results and other things, a few things we have done to just do the sanity check, right? We have 2 different studies, study 1, study 2. We have 2 different doses. We wanted to see is any clusters are observed here. We didn't see any clusters, either the study-wise clusters or the dose-wise clusters. So it's pretty much all over the place. Some kind of artificial clusters. We have seen this, but we do not know what it really means. So then next step, what did is when we're looking into the data analysis, it started with the univariate analysis, right? Simple statistical methods. Correlations, for example, correlations between individual endpoint versus the 162 parameters we had, and wanted to see is there any particular variable is significantly correlated with any of those 5 different endpoints, right? And then for each and every variable or the parameters, 162 plus parameters we have used, we quickly looked and created a box plots and tried to find out, do we see any huge variation is present in those 2000, even we did the mean and standard errors, any huge variation is present, any outliers are present. So this is again to do some sanity check, and then we done quickly a t-test and the chi-square test, etc., etc. See there is any variables which are simply visible and they are, without getting into the model, they are giving us some kind of a statistical difference separating the responders and non-responders based on the endpoints. Yes, there were some SNPs which were suggesting they were separating the doses 150 and 300. There are some SNPs separating the effect. There are 4 of them. And similarly, a particular biochemical enzyme, the gamma glutamyl transferase, was also had some differences between responders and non-responders. So these are the initial observation. But what we have done is we have moved ahead and built a model. Here, just wanted to convey one more time here. 6 different teams. Each team, if you look at it, these are the different machine learning models, right? So each team, a particular team has picked one model. The other team has used about 6 different models. So we have given the freedom to anybody, to everybody to use whatever the model they want. End of the day, they have to use the data with their own way, test and train and establish the accuracy, sensitivity, and specificity. Once that is done, they should be taking these final data set. This is again another anonymous 800 model data set, and they need to run. The reason we use the 800 is here they did different things. So we wanted to bring the commonality across the board. That's the reason we gave 800 to finally to verify. Okay. So just a snapshot, different models has been used here on the rows. The different teams, strategy, we call it strategy 1, 2, 3, 4, 5, different, they use the different, for example, strategy 1, all data model, they only use this and support vector machine. A strategy 2, that particular team had about, within that there are 3 different variations, they use the tons of ML methods. So bottom line is everybody has a free hand in whatever they want to do. But ultimately, once they build the model, when we gave these 800 models set, and when we say that you have to test, this is the 160 random test set. This is 800 to build the model, same way they have built in the previously, 160 to test, and then we predicted the accuracy, sensitivity, specificity from all models. Now, the next step is, what are those? What did we get? Right? What are the potential features and the signature we got from different models? Right? These are some of the top of features related to this particular study, the case study. Some are biochemical ones, some are the SNPs. So it's some baseline information. So it's a varied from different angles. So, and then since we have 6 different teams were used, did build the model, 2 teams at some point they dropped, saying that, my model is not doing any good, so I'm trying to abandon. So then we ended up with 4 different outcomes. So then when we looked into it, is any of the feature which is present in all 4 models came out as an outcome from 4 different models. What are the other things came out from a 3 and a 2 and a 1? We kind of established the frequency. Then we started looking into it. What are those? Went each and everything and trying to establish the scientific justification. Out of that, there are 4 known biomarkers from the literature which appeared out of those, multifold or multi-pronged approach. There are 8 novel biomarkers, and out of the 8 SNPs which came out on the top, the 4 are known ones, which are known to have through the information of the known biomarker, and another 4 were a novel biomarker. So that way we have established the different. Then the next major step is how do we validate it, right? So we went ahead and looked into, spent quite a bit of a time understanding the biological relevance for each and every feature. How these parameters or the features are relevant, and then we established a certain justification, right? And for each and every one. Then some of these could be a novel potential marker. Some of them have directly has the literature support. So I'm not giving the whole lot of details, apologies for that, but just please bear with me. This is just to showcase that, how we have done. And then some of the SNPs, we kind of know where they are, and now we kind of mapped on the overall pathways and the role in terms of how they may play a role in a responders and non-responders. So we did this. These are the 5 which has a direct literature support. Then there are another 4 SNPs which doesn't have a literature support, but certainly may be useful if validated down the road as a novel and potential markers. Okay, so that's outcome came, and from that outcome, what is the final recommendation? How those markers were useful in a practice, right? So, in a key, when we look at the key conclusions, there were altogether 21 features we have identified. That the 9 features has a strong evidence of being associated with the drug's mechanism of action or the disease progression. 3 features are linked to therapy or disease indirectly, right? Then the 4 genotypes or the SNPs are identified as a novel, hence this could be a potential new things. In addition to this scientific evidence, we also come up with a couple other recommendations saying that some of these 4 clinical parameters can be recommended or useful as a potential inclusion criteria when we design the phase 3 clinical trials. Increased expression of the 4 targets also could serve as a potential inclusion criteria to identify the right patient who are responders. 3 hematological parameters, we also felt that these are more meaningful, specifically looking into this kind of individual patients with the interleukin antagonist. So in addition to that, we observed some kind of a comorbidities within when we in the individuals we have tested. So based on the comorbidities, we recommended a combination therapy. And whoever is the comorbid patients are there, it is better to include and introduce another drug in addition to what is the drug clinical candidate we are using in this particular study. So all in all, this exercise was useful to identify the markers, also to help the design the clinical trial design in a better way for in the future, and then opened up the new avenues for the combination therapy. So this is the one case study. Next case study, I will go a little quickly. I know we may be running out of time, but this case study is very interesting one. This is the CAR-T trial. We all know CAR-T trials is very expensive, very useful, and lately, there is so much of a focus and effort is being there. In this particular left side of the slide talks about specifically comparing to the 2012 to 2024 or 2019 and 24, how the CAR-T trials are progressed, how many number of trials are increased, etc. Pretty much talks about in a nutshell, the importance of the CAR-T trial. And on the right side, specifically when it comes to the CAR-T trials, one of the most important thing is whether an individual need to be an inpatient or an outpatient. We will have once we take the CAR-T cell therapy, the most important thing is there is a side effects, right? You may have a cytokine release syndrome. You may see the neurological event. It all depends on whom we are giving this therapy or the treatment. So the inpatient burden, first of all, the CAR-T treatments itself is expensive one, right? Then the inpatient, if you put the individuals in an inpatient, the burden is going to be quite a bit of a high. There is so much of a cost is involved. Hence, our partner felt that can we build an algorithm whereby looking at the initial parameters of the individual, can we predict at a given subject is going to be a potential inpatient candidate or an outpatient candidate? I think that was the major question being asked in this particular case study, right? So with that background, so our goal was in this case, we had about 108 patients from a phase 1 clinical trial, 321 parameters across the 3 different time points. We have taken that pretty much the same approach, data formatting, algorithm building, outcomes, and more or less the same challenges, like any other AI/ML method. But the end goal is identify or to classify the patients who are, whether they are supposed to be an inpatient or outpatient. That's the outcome we were expecting, and we are hoping that certain markers would come out and classify those. For that, there, like I said, we have three different doses. I would say collection points. Baseline, it is day 0. Day 1, that is after taking the CAR-T therapy, then a day 2. So the hope is, if we can able to identify using the day 0, there is nothing like it. The parameters, and say that if you have a so-and-so parameter is high, the baseline levels, that particular subject is prone to have a side effect. Hence, that individual going to be an inpatient. So if that is not possible at a day 0, we can at least see on a day 1, because by the time the drug kicks in to the certain extent, and the side effects may kick in. So hence the at least a day 1, if not a day 2. So that is the reason we wanted to get build the models separately for the day 0, day 1, and day 2. So those are the three different time points. Data is taken, the models are being built. So we did discuss about this on the left side, and these are the different parameters. I've just provided the representatives of it. What are those? But it's a different class or clusters of these pharmacodynamic parameters, product characteristics, the baseline parameters, lab measures, etc. All of those things, the 321 parameters was collected day 0, 1, and 2, as I mentioned. Then another most important factor is the individuals, right? When the subjects are taken this in a phase 1 clinical trials, we have defined those into outpatient definition 1, definition A, definition B, and definition C. What it means is, if a definition A means if so-and-so things, the CRS criteria is this, there are no neurological events. So we kind of separated it out, who is who. So we also wanted to build the models based on the individual's group of subjects cohorts. Definition A is a cohort with a certain criteria. B is another cohort. C is another cohort. See, these three different cohorts and three different time points, 0, 1, and 2 days, right? So it's 3 by 3 matrix. So there are a total 9 different potential models are built. Same process, data processing, data building, model interpretation has been done. And when we looked into the individuals, A, B, and a C, definition A, B, and C, these are the features which are cumulative features, not slotting into these different circles here, but I'm just representing as the cumulative feature for proprietariness sake. And these came out as a potential feature that was able to identify the inpatient and outpatient classification. If you look at, that's what we wanted to present here. So, and then finally, our goal was, once we build this model, here this model is built, and then next time when we recruit a patient, we collect those 320 parameters from the patient and examine this individual patient profiles against the model built, and say whether this particular given a patient is supposed to be an inpatient or an outpatient. That particular platform was used to separate it during the phase 2 clinical trial recruitment. So that was the overall conclusion. We have made the platform as built and given it to our partners. So Yep. With that, I'm almost reaching to the summary here. So in all, what I wanted to conclude is patient stratification is very much important to have a right medicine for the right patient, right? And this will certainly play a major role in the overall, clinical trial practice going forward. To build the right model, I must say that we need to have a right data, right? Great, so start with the right data, but right data still need a right processing, and we need to make it ready for the modeling. And then once the model need to be built, and again, there are the tons of models out there. It's a question of what is the right model. There is so much of iteration is required with this when slicing and dicing the data. And end of the day, models are models, right? Models will spit out a certain outcomes. But unless those outcomes are supported by the scientific hypothesis, scientific justification, and then only it will further pave the way to validate those biomarkers. Otherwise, if you don't have a scientific justification, it doesn't make any sense. And we can just say this model gave something, right? So with that, like to conclude that, the data processing is the most important. It occupies about 40% of your effort. Modeling is modeling. Once the good data is there, it requires a small percent, 20% of effort. And the last, it's great to have these artificial intelligence, but biological interpretation require the human intelligence. There is no model which will automatically interpret the things for us. So the scientist has to be. So artificial intelligence followed by the human intelligence is required in order to build a patient stratification overall models. So with this, we'll stop. Thank you very much, everyone.
Thank you, Nandu. With that, the floor is now open for questions. As I'd mentioned at the beginning, please drop in your questions in the Q&A box so we can start answering them. In the meanwhile, while we are waiting for questions, Nandu, were a few questions that have already been posed. So the first one, how do you trust the features that are predicted by the models in the first case study? Is corroboration and overlapping features the only way to go?
Okay. So if you look at the case study 1, we deliberately use the different models, right? When we use the different models and when we gave the free hand to the individuals, obviously, whatever the data goes in, whatever the model we have used, some variation is very much reasonable. But if the question is, end of the day, what is that coming out? If the outcome or if the parameter is the most significant and it's a real one, it should come no matter which model you use. And in our case, the certain parameters, certain features are present in multiple models. Hence, those features are without a second thought, we must use it. But the ones which are present, the features in one or the other model, we certainly look into further, put an extra lens with the science and other things and start using those as appropriate with the proper justification and the validation. But I would say that variation is the common, but valid scientific hypothesis and justification is very much required.
Yep. As a follow-up question to that, Nandu, another question in the box. When you had the 6 teams approaching the same problem, were they allowed to talk to each other, or were they siloed? Were they blinded from what the other team was doing?
So in the initial days, right? So where we are processing the data, it was collaborative effort. Everybody chipped in and say that, okay, this particular variable, we should remove it. Or this particular variable is similar to the other variable, so we should, combine these things. Or for some of the labels, 5 labels we created an artificial another endpoint. So there was quite a bit of until we massage the data, until we process the data, everything was a collaborative. And once the data processing is done, everyone was doing on their own. But of course, everyone was coming on alternative day and presenting their own model, because I may be using the random forest, somebody may be using something else, but everybody was presenting how well that is accuracy, sensitivity, specificity. We literally did not lock them in the rooms, but certainly gave an opportunity for individuals to do what they want to do in their own way.
But you would say that they were allowed to inform each other's process as they went through.
Absolutely, absolutely. They were talking to each other, and I'm using this model. See, it's giving the good results. Blah blah blah. They were talking to each other, but the process and whatever they wanted, the way they wanted to use, is totally different.
All right. The next question is. Were the features that were predicted by the univariate analysis, which was initially done in the first case study, did they show up in the machine learning analysis as well after all the 6 different approaches were used?
Yeah. Thanks, Simona. So, see, I mentioned in univariate analysis is initially to just look at the sanity check part of what's happening. Is there any visible parameters or the variables that are clearly separating through correlation, t-test, or the square, chi-square, etc., and some biochemical parameters, some SNPs were significantly different. And obviously, some of them came in the final, some did not. So to me, it's okay to see what's univariate in the analysis, the things are coming out. But I would, when we're building the model, I see that there is a lot more holistic approach, a lot more weightage would come in. So I would trust more on the ones with model rather than a simple univariate analysis, but I would not ignore those also. I would take even the features came out of the univariate analysis and try to see if there any scientific justification is out there that is more meaningful. So yeah, I would certainly, there is an overlap, but there is an exclusion too.
All right. One other question then, does patient stratification always have to be retrospective, where we are analyzing retrospective data to build these kind of models? Or can it be real time as well as someone is going through the clinical process, through the clinical trials?
Right. Thanks, Simon. I'm sure, this question came to your mind over us, but, it's because we presented 2 clinical trial-oriented case studies, right? So in this particular ones, yes, obviously, we have used the previous data to build the right models. And, but, when we in a preclinical stage, we don't have to use the retrospective thingy. In a preclinical stage, we typically use the cell lines and look into whatever the compound we have, look at the IC50 values or some other phenotypes like apoptosis, etc., and then try to build the models and have a sensitive and resistance things. Try to build the model. There mostly those is the prospective in the preclinical area, but in a clinical, when we look the clinical thing, most of the times it is a kind of a prior data is required.
Yep. Another question, Nandu. What are the potential regulatory hurdles in patient stratification strategies and models that you have encountered?
I'm sorry, what are the potential?
Regulatory hurdles?
Well, we haven't, in terms of doing this, everything was, since we have worked with our partners, everything was anonymized for us. And so we do not know. It's all, not necessarily regulatory related things that we handle per se. We consider this as the data, hand it over to our partner, and it is up to the partner on how to validate, how to make it more regulatory oriented, compliance and other things. But in our case, we didn't really look into from a regulatory perspective. We consider this as the data science problem more than anything else.
All right. I would urge people to, most of you have been allowed to speak. So if there are any questions, if you're not dropping them in the Q&A box, please feel free to unmute yourself and ask anyone on the panel any questions. Again, thank you to the San Francisco Computational Biology Group. Anyone here who would like to join it, please feel free to drop in a message to Russell. He will be happy to give you some information on how to join.
Good. Thanks a lot, Simona. I'll stop sharing and you can take it from here.
All right.
I actually have a question for Nandu about the outpatient classifier. So during that analysis, were you able to take into account potential differences in efficacy, for example, in populations of patients that experienced an inpatient event versus an outpatient event? Because one possibility, I guess, would be that you could inadvertently build a classifier in which there was unfavorable PK/PD or something like that for patients, and kind of undermine the trial, if you know what I mean.
Yeah. Russ, are you asking about the case 1 or case 2? Case 2 is outpatient and inpatient.
Yeah, I was specifically thinking about the case 2 one, but just in general, I wonder if you have thoughts about that possibility.
In this particular case, Russ, since it was the phase 2 data, the efficacy wasn't considered as an endpoint here. It was mostly, from a day 0, 1, and 2, we were focused mostly on the adverse oriented, those we are considered as the endpoints and looked into it, but not the efficacy.
Yeah, I think, Nandu, you meant that the data was phase 1 data. It was more focused on efficacy initially, safety initially, before going into phase 2, where efficacy would be the prime concern.
That's what I mean. Yeah.
Does that answer your question, Russell?
Absolutely. Thank you. Yeah. I also, just from the perspective of someone who works at a company that's interested in human genetics information, I noticed that during the data processing section in the first case study, you talked about disregarding ancestry and racial reported information. Obviously we're kind of interested in many diseases that appear to have ancestry as a fairly important covariate. So I wonder if you looked into that as potentially incorporated into a model or, further providing information.
Right. It's a choice in this case, Russ, more than anything else. But, obviously in certain other cases, the race may, literally have a huge weightage in terms of how they respond because of the genetic composition or other things. But in this particular case, it was just a drop. But I totally agree with you. There is no reason to drop it. We could have kept it and see if there is something that is going to have an impact also.
Yeah, and I think it has more of an implication based on the therapeutic area. In this case, we were looking at autoimmune diseases, but if we were specifically studying, looking at studies associated with, say, rare diseases where genetic biomarkers are probably going to have far more weightage than they would, ethnicity-related markers would have far more weightage than they would in an autoimmune disease, then yeah, that would be the prime sort of feature to look into.
Yeah.
All right. So there are no further questions. We'll just give all the participants another 30 seconds to drop in any further questions. in the box. Post that, if there are any questions dropped in, we will try and answer them via email. With that, I will now hand this session over to Adi and Russell to sort of close out, and then we'll take it forward from there. Adi?
Yeah, sure. Thanks, Nandu, Simona, and appreciate Russell once again on setting this up. So having said on some of the approaches Nandu and team were taking, I thought we'll also give a quick background on the company for the folks who might have joined late or might have not known or heard of Excelra as well. So I thought we'll do that and maybe also recap on some of the features and benefits on the South SF CB forum that you run, and maybe a little bit on Maze. Russ, if you want to share some of your responsibilities and work, that would be helpful as well.
Sure, I'm happy to do that. Yeah. Sorry, are you going first or am I?
You can go ahead. You can.
Sorry.
No worries.
Yeah. Well, as I mentioned before, like the South San Francisco community is really this collection of scientists in the Oyster Point, Lower Peninsula kind of area of San Francisco who are engaged with various companies that really are just interested in exchanging ideas and building a social network that helps support our various tasks and things like that. And then Maze Therapeutics, of course, as I suggested, is a human genetics company that's interested in developing disease, basically using human genetics information to better identify disease targets. And I don't know, maybe we can talk offline another time if you're particularly interested in the details of what we do there. Sorry, I feel like I interrupted you, Adi.
No worries, no worries. So maybe Simona, you can go on the next slide. Yeah. So just to give everyone an overview on what we do, who we are. So we have been in the business for the last 2 decades, trying to curate data. We are focused only on the life science and the pharma domain, and the only single focus at Excelra is to empower drug discovery and life science innovation for the companies using our tools, technologies, and resources. We are headquartered in Hyderabad, city called Hyderabad, out of India, and our business is spread across the globe. 60% comes from the US, 35% Europe, and the rest 15% out of Asia. And little sheer presence being in India, we do support a little bit of local companies as well. And since it's a San Francisco-based event, of the 60%, 30 to 35% of the business comes out of the West Coast as well. What we do data wrangling. What we mean by that is we try to curate data. We integrate data in various formats or machine readable formats for various partners, and try to harmonize that into a database if people are building some internal sources or data lakes that they will. The second piece is the data engineering, where we build various platforms, build tools to integrate data, and try to automate this or build enterprise-wide applications, right? So that's on the data engineering piece. And then data analytics, where today Nandu and team presented. So it's more on the data science or bioinformatics at the core, if you will, to address various scientific questions. So in a nutshell, that's what we do. And this is done by our amazing team, pool of 500 scientists, all FTs based out of India. And we have diverse skill sets across discovery chemistry, biology, bioinformatics, data science, clinical pharmacology, and right up to real-world evidence. And of course, the underlying technology layer on the IT and informatics side. So that's a bit about us. And next slide will give you an overview of the portfolio. I'm not going to go through each line item. But if you see right from the left to the right, if you scroll on the screen, we start right from early discovery, cover the translational piece and clinical piece, some of those elements that we were discussing today. And then the value evidence element, which is our growth strategy to cover the entire drug discovery spectrum. And if you see technology, that's the underlying layer across various aspects. We can support that too. And we'll circulate these slides as well, along with the recording. And feel free in reaching out to us if there are any questions and if there are ways we can help you.
Yep.
Yeah, if it was person, I thought was maybe beer time or maybe good soft drink, or summer is right now, so a lemonade would be good. But currently it's midnight for us, past midnight, so trying to stay awake and drink some coffee.
It's a sleep time, Adi.
That's why I have a coffee mug in my hand.
Okay, thanks everyone.
Thank you guys so much. This has just been wonderful.
Thank you everyone.
Thank you again, Basil. And thank you
Vasan,
to
for hosting us,
everyone
and
who
thanks for
joined
all
in.
the participants.
Yep.
Bye.
Thank you for being with us through the last hour and a half.
Of course, yeah. Please go get some sleep, because I can't imagine giving a talk like that at midnight. That's very impressive. After about 7 o'clock at night, I have a lot of trouble thinking straight, but I have 2 children, so.
Yeah.
Thanks.
Okay, thanks, Ross.
Thanks very much.
Thank you again, and have a great
Stay safe.
weekend, everyone. Bye.